Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the Pearson Edexcel International A-Level Biology Unit 5 for October 2023. Let's begin with the first question. In question 1, the image shows a section of a recombinant DNA molecule. So they say which enzyme is used in the production of recombinant DNA. To produce recombinant DNA, we use restriction enzymes, so the answer should be C, restriction endonucleases. Then they said DNA is present in bacterial cells. Where is DNA found in bacterial cells? Bacterial cells have no nucleus, so DNA is going to be found in the cytoplasm or in the plasmid. The answer should be C. So next they say which bond joins nucleotides together in a single strand of DNA. Because they said single strand of DNA, this should be a phosphodiester bond. And then describe how genes can be switched off by transcription factors. These are proteins that bind to the specific sequences of DNA, blocking the binding of RNA polymerase. So this brings us to the end of question 1. Let's continue to question 2. In question 2, the photograph shows the eyes of a meerkat. They say describe how the pupil of an eye dilates and contracts in response to changes in light intensity. So here I'm going to discuss light intensity changes, maybe high light intensity as well as low light intensity. We know the pupil of the eye is going to be varied because the muscles of the iris are going to contract or relax. So I said light intensity is detected by photosensitive cells in the retina, and impulses are going to be generated and sent to the central nervous system through the optic nerve. The motor neuron returns the response to the muscles of the iris, and we know the iris contains antagonistic muscles that control the diameter of the pupil. In bright light, circular muscles will contract, radial muscles will relax, and the pupil is going to constrict. And in dim light, radial muscles will contract, circular muscles will relax, and the pupil diameter is going to be dilated. In part B, photoreceptors stimulate nerve transmission in the eye. Which of the following pairs of molecules forms the photoreceptor in an eye? The answer should be C, opsins and retinol. And next they say which of these drugs would increase nerve transmission. So when we look at the possible answers, lidocaine is not used in increasing impulses because it blocks nerve impulse transmission. So any answer having lidocaine is going to be ruled out, and therefore the answer is going to be B, ecstasy as well as nicotine. In direct sunlight, the diameter of the pupil was 4 millimeters. In complete darkness, the area of the pupil was 38.75 millimeters squared. Calculate the increase in diameter of the pupil when moving from direct sunlight to complete darkness. So I began with this answer here because in direct sunlight the diameter is 4. In complete darkness they've given us the area. So diameter, using this formula here, area is pi r squared. This is the area of a circle. So radius should be diameter divided by 2. Substituting for that, I got area is equal to pi times diameter squared over 4. And making diameter the subject, I got diameter is equal to the square root of 4 times area over pi. So I can substitute the area, use the pi in the calculator and find the diameter. So substituting this area here, I have to put the square root here like that. So substituting here the area and then 4 divided by pi and the square root, I got 7.02. This is going to be the diameter in complete darkness. So the change is going to be the bigger diameter minus the smaller diameter, which gives us 3.02 millimeters, and that is going to be the answer. So this brings us to the end of question 2. Let's continue to question 3. In question 3, acute disseminated encephalomyelitis causes the destruction of the myelin sheath around neurons. The destruction is a result of inflammation. Lesions form in the brain where the myelin is destroyed. The image is a magnetic resonance imaging scan of a person with ADEM showing lesions in the brain. So they say lesion A is spherical and has a diameter measuring S to T. We get 1.97 centimeters. They say calculate the volume of lesion A. They want you to use this formula. So because they've given us the diameter, radius is going to be diameter divided by 2. And I got that divided by 2 giving me that. So the volume is going to be 4 divided by 3 times pi. The pi I used here was the calculator pi times 0.985 power 3. And rounding out to 2 decimal places, I got 4.00 centimeters because the answer was required to be in 2 decimal places, so that was my answer. Next, they say calculate the actual width of the lesion labeled B between point X and Y. Give your answer to two significant figures. When I measured XY, I got 0.55. So here I use the ratio of these two. ST to XY is 1 to 0.458. So I can use the actual values. The actual XY is going to equal to 1.97 times the ratio, which is 0.458, giving me 0.90 centimeters. So that is going to be the answer. And then they say an MRI scan can be used to diagnose a brain tumor. Suggest why a dye might be used during the MRI scan. A dye is used to improve visualization, so I say it so the image can be enhanced or can be made clearly visible. In part C, a person with ADEM has problems with walking. Explain why ADEM causes problems with walking. Remember, this condition removes or decreases or breaks away the myelin sheath. It means nerve impulse transmission is going to be slowed. So I say due to loss of myelin, nerve impulse transmission slows down. Fewer impulses reach the legs. Muscle contraction is going to be affected, so the person is going to walk slowly. So this brings us to the end of question 3. Let's continue to question 4. In question 4, they say the mean resting heart rate varies between species of mammal. The table shows the mean resting heart rate and mean lifespan for 7 mammals. So we have ranging from the mouse to the whale, the corresponding resting heart rates as well as the lifespan. So we can see as the mean resting heart rate decreases, lifespan increases except the anomalous one, which we see here. They say describe two conclusions that can be drawn from this data. Like I've already said, the higher the mean resting heart rate, the lower the mean lifespan. The mouse, this one here, had the highest mean resting heart rate and the shortest mean lifespan. Also, the humans have the longest mean lifespan. However, the result is anomalous in the trend. In part B, they say an electrocardiogram ECG is used to identify abnormal heart rhythms. Trace A shows a normal heart rhythm and trace B shows an abnormal heart rhythm. They say what is the heart rate shown in trace A? So for trace A, remember here they've given us the scale as 4 small squares correspond to 1 second. So here it means 5 small squares, which give me 1 full heartbeat, correspond to 1.25 seconds. So here 1.25 seconds correspond to 1 heartbeat. Converting this to minutes is going to be 1.25 divided by 60 minutes. And therefore in 1 minute we're going to have 1 times 60 over 1.25, which is 48 beats per minute. The answer should be B. Next, they say describe two differences shown between trace A and trace B. So we can see, look at the QRS wave is different. We can see this peak is shorter in comparison to that. There are so many differences. We can see this one here is lower, or it's kind of more deeper in comparison to that. So I said B has a longer QRS wave duration, B has a shorter T wave, and A has a shorter P wave. In part C, cardiac muscle is unable to regenerate or repair itself. Mammalian heart muscle can beat only a certain number of times in a lifespan. Humans have mean resting heart rates of 72 beats per minute and a mean lifespan of 8 years. Suggest 2 reasons why humans can have a different lifespan from other mammals. This is mainly because humans have technology and there is a conscious decision to make life changes. So I say they have more technology on how to take care of diseases with vaccination, medication, and surgery. They can also alter their lifestyles if progression of disease, like CVD, is detected. The next state why cardiac output provides more information than the resting heart rate. Of course, cardiac output is equal to heart rate times stroke volume, like the volume of blood that is pumped out per specific beat. So this shows the efficiency of the heart. The next they say an African giraffe has the longest stride of all land mammals and can run at a speed of up to 60 kilometers per hour. Describe how the nervous system affects cardiac output to enable the giraffe to run at this maximum speed. So this is attributed to chemoreceptors that can detect changes to the concentration of blood and send nerve impulses to the brain so that the heart rate is increased. So I say to achieve maximum speed, the cardiac output increases on running. More carbon dioxide is produced, leading to a drop in pH. Chemoreceptors in the blood vessels detect the change and send impulses to the medulla's cardio control center. Impulses from the control center through the sympathetic nerve arrive to the sinoatrial node. More depolarization occurs and the cardiac muscle contracts with more force to increase the cardiac output. So this brings us to the end of question 4. Let's continue to question 5. In question 5, dopamine, serotonin, and acetylcholine are neurotransmitters in the human nervous system. Describe how a neurotransmitter allows an impulse to be transferred to an adjacent neuron. Here we're talking about what occurs at the synapse. So I said, arrival of an impulse in the presynaptic neuron causes calcium ion channels to open. So more calcium ion will enter the presynaptic neuron from the synapse, and the calcium ions bind to the vesicles containing the neurotransmitters, causing the vesicles to fuse with the presynaptic membrane. The neurotransmitters are released into the synapse, and they diffuse through the synapse and bind to the receptors on the postsynaptic neuron. This causes sodium ion channels to open, and the propagation of an impulse in the postsynaptic neuron occurs. In part B, they say the diagram shows how serotonin is produced from tryptophan. So we have tryptophan converted into 5-HTP by enzyme A, and that is going to be converted into serotonin using enzyme B. So they say enzymes A and B are found in the Madagascar periwinkle. Medical serotonin can be produced by genetically engineered E. coli bacteria. Describe how E. coli could be genetically engineered using the Madagascar periwinkle to produce serotonin. So in genetic engineering, we begin by isolation of the specific genes, and then we find ways of using restriction enzymes to cut the specific genes out of the DNA as well as the plasmid before we can insert the genes into the plasmid. So I said isolate the genes for enzyme A and B from the DNA of the Madagascar periwinkle, use the stem restriction enzyme for the periwinkle DNA, and for the plasmid, this leaves complementary sticky ends. Join the periwinkle genes to the plasmid using DNA ligase, and insert the plasmid into the E. coli bacterium, grow the E. coli in a fermenter, and purify the produced serotonin. In part C, they say the genetically engineered E. coli can grow in a liquid culture. The concentration of tryptophan and serotonin in the culture were measured. The graph shows the results. You remember tryptophan is going to be the reactant and this is the product. So we expect as the reactant decreases, the product is going to be increasing if we give the experiment time. So they say discuss the changes shown in the graph and use the information in the question to support your answer. So I said serotonin concentration increases as the tryptophan concentration decreases for the experimental time. The changes, meaning decreases and increases, are faster before 30 hours and they slow down after 30 hours. We can see, of course, there is going to be a faster increase, but around here we see a slower increase and slower decrease. In the reaction, tryptophan is converted to serotonin by the enzyme produced from the genes of the genetically engineered E. coli. So this brings us to the end of question 5. Let's continue to question 6. In question 6, some processes in plants and animals are under the control of chemicals. Which row in the table shows the effect of an increase in adrenaline in a mammal? When adrenaline is released in a mammal, the breathing rate increases and the heart rate increases, so the answer should be C. Next, they say nicotine is a drug found in tobacco products. The effect of changing the concentration of nicotine and adrenaline on the mean heart rate of Daphnia was investigated. The diagram shows a Daphnia. We can see its heart, and that is going to be the Daphnia. So they say the results are shown in the table. So here we can see nicotine concentration. It decreases as you go down here, and the control has no nicotine. So here we are measuring the mean heart rate as the concentration of nicotine increases, and here as the adrenaline concentration increases. So here we're going to have the highest nicotine and the highest adrenaline. So we can see the heart rate is high, but it's actually not the highest. The highest heart rate in the non-control sample is here. Of course, we have the highest in the control, but I'm not considering the control. In the non-control sample, this is going to be the highest. That means lower nicotine and high adrenaline cause a high heart rate. So we can see adrenaline is going to be a stimulator. And here we can see the lowest heart rate is in high nicotine, lower adrenaline. So it means nicotine is not a stimulator. The that means nicotine is a depressant. So here adrenaline is going to be a stimulant and nicotine is going to be a depressant. They said deduce the effects of combining these chemicals on the heart rate of Daphnia. I said with or without adrenaline, the higher the concentration of nicotine, the lower the mean heart rate. The mean heart rate increases as the adrenaline concentration increases. The lowest heart rate is 81 bpm with 0.001 millimole per decimeter cubed adrenaline concentration and 1000 millimole per decimeter cubed nicotine concentration. And again here I'm not writing about the control samples. The highest heart rate is 151 bpm with 0.1 millimole per decimeter cubed adrenaline concentration and 0.001 millimole per decimeter cubed nicotine concentration. Increase in adrenaline concentration without nicotine increased the heart rate, and at 0.1 millimole per decimeter cubed adrenaline concentration without nicotine, there is no measurable heart rate, meaning the organisms could have died because there was no detectable heart rate. In part B, they say which is a description of a gibberellin. Gibberellin is a stimulant, so the answer should be C. It's an organic compound that stimulates the action of transcription factors. And next they say the quality and length of daylight acts as a signal to initiate and regulate flowering in plants. This involves phytochromes and the production of gibberellins. In an investigation, a long-day plant and a short-day plant were exposed to light for different lengths of time. During dark periods, they were exposed to flashes of red infrared light. The diagram shows the results of this investigation. So we can see for plants there was flowering here, and here. And for short-day plants there was flowering here, here, and here. We observe that flowering in long-day plants occurs if the day length is greater than 12, maybe between 12 to 14 hours. If the day length is not that great, then we need to include red light to stimulate flowering, because we can see every time the last exposed light is far red, there is no flowering. We can see here red is the last one, there is flowering, and far red there is no flowering. So either the critical light length is going to be longer, or we can expect red light causing the stimulation. For short-day plants, for flowering to occur without the red light, we need a period of less than 12 hours of exposure to light. We can see this is 12, so the light exposure for flowering to occur should be less than 12 hours, or it should be when far red was the last light they exposed to during the dark hours. So flowering occurs here as well as here. And next they say the graph shows the results of an investigation into the effect of gibberellin concentration on the flowering of another type of plant. So here we can see the mean number of flowers on the vertical axis, and on the horizontal axis we see the concentration of gibberellin. As the gibberellin concentration increases, the mean number of flowers increases until about 1.5, and then we can see increase in gibberellin concentration leads to a decrease in the mean number of flowers. So here they say discuss how phytochromes and gibberellins affect flowering in plants. Use your own knowledge and the information in the question to support your answer. Like I've already gone through the question, I explained some of the results. So here I said, from the investigation on light exposure, short-day plants require less than 12 hours of daylight to flower. They can also flower when exposed to short periods of far red light during the night. Exposure to red light does not cause flowering, meaning if red light was the last light they were exposed to, they will not flower. For long-day plants, flowering occurs when exposed to light of about 14 hours a day. Also, if exposed to red light, exposure to red light, then far red prevents flowering. Phytochromes red and phytochrome far red absorb red light and far red light respectively. Here it's because this absorbs that and that absorbs that. High PFR or low PR leads to flowering in long-day plants. A low PFR and a high PR leads to flowering in short-day plants. The higher the concentration of gibberellin until 1.5 micrometer per decimeter cubed, the higher the number of flowers. After 1.5 micromole per decimeter cubed, increase in gibberellin concentration leads to fewer flowers. Gibberellin could promote flowering at low concentration and inhibit at high concentration. Gibberellin causes the oleuron layer to secrete amylase. They could also act as transcription factors affecting the expression of some genes, and this could be linked to flowering. So this brings us to the end of question 6. Let's continue to question 7. In question 7, When antagonistic muscles contract, they move skeletal bonds. Energy is needed when a muscle contracts, which row in the table shows the energy used in muscle contraction. In the table, to break the actin-myosin bridge, we do need energy. To remove calcium ions from the sarcoplasm, yes, we do need energy. Release of ATP and PI, this does not require energy, so the answer should be B. Next, they say describe the sliding filament theory of muscle contraction. Here we have to talk about troponin, tropomyosin. We talk about the myosin, the actin, as well as the ATP and the movement of the myosin head so that the sarcomere is going to be shortened. So I said when an impulse arrives at the neuromuscular junction, calcium ion channels open and calcium ions are released from the sarcoplasmic reticulum. The calcium ions bind to troponin, causing a change in conformation, allowing the exposure of the myosin binding side on actin. Myosin binds to actin, forming an actomyosin bridge. Then PI is released from the ADP-PI. Actin is pulled over myosin, shortening the sarcomere. Then ATP binds to the myosin head. The actomyosin bridge is broken as ATP is hydrolyzed into ADP and PI, providing the energy required to move the myosin head back into its original position. In part B, they say two types of ostrich, the South African black and the Zimbabwean blue, are found in South Africa. Ostrich muscle is a source of dietary protein. Scientists compare the mean masses of three muscles from the ostriches. So here we can see the mean values that were given with their corresponding standard deviation. I calculated the highest and lowest by adding this to that to give me that and subtracting this minus that to give me that. So I calculated for each and I realized there was overlap here, there was no overlap here, and there was no overlap there. So they say step two conclusions that can be drawn about the mean mass of muscle shown in the table. Again, based on the information we have, muscle T has the largest mean mass and muscle F has the smallest mean mass. The Zimbabwean blue ostrich have great muscle mass for all muscle types. Their values are higher in comparison to that. The differences in the mean muscle mass in G and F for the two ostriches are significant because there is no overlap. They say comment on the validity of conclusions drawn from the data for these two groups of ostrich. When they talk about commenting on the validity, we have to talk about if there is overlap or not. We have to talk about the methods used to collect data, talk about the sample size, and so on. So I say there is overlap in muscle T for the two ostriches, so no significant difference in the results. Muscle F has no overlap for both ostriches. It has the lowest standard deviation, so the results are valid. The sample size used in the Zimbabwean blue is small, so the results are not reliable. Next, they say calculate the percentage difference in the mean mass of muscle G of the South African black and the Zimbabwean blue ostrich. Use the equation. Percentage difference between A and B is that. They want us to give the answer in two decimal places. From before, I'm taking the South African one to be 0.59 because we're using muscle G, and the blue ostriches to be 0.74. This is from the table. So if this is A and that is B, the difference is going to be 0.59 minus 0.74. Divide that by 0.59 plus 0.74, divide by 2, and then multiply everything by 100. I got negative 22.556, which is approximately 22.56%. The hypothalamus is involved in maintaining a steady internal temperature in the ostrich. Describe the role of the hypothalamus in maintaining a steady internal temperature. The hypothalamus is going to detect the change in temperature, and then impulses are going to be sent out to respective organs in order to cause a required change. So I say thermoreceptors in the hypothalamus detect a change in the temperature of the body. Then the thermoregulatory control center receives impulses. Then impulses are sent via the sympathetic nerve to the effectors in the skin, liver, and muscle. A response is carried out that leads to a corrective change. If the body temperature is increased, then vasodilation occurs, metabolic rate slows down, and so on. So this brings us to the end of question 7. Let's continue to question 8. We're going to begin question 8 by showing you the scientific article. If you look at my channel, I already went through this article, so I'm just going to go through this showing you. I'll be silent, and then after we can go to the questions. Question 8 says the scientific documents you have studied are adapted from articles in New Scientist entitled Your Second Skin, May 2022, and Anatomy Fascia from ResearchGate, December 2018. The first question says give the meaning of the term sensory organ. A sensory organ is a group of tissues working together to detect a change in a stimulus, so that's quite easy. Next, they say fascia is a type of connective tissue. Use ticks and crosses to complete the table comparing types of connective tissue found in the human body. So fascia contains collagen, of course. It's going to be rich in nerves based on the article, and it surrounds muscles. Ligaments connect bones to bones, so they contain collagen. They do not have, or they are not rich in nerves, and they're not surrounding a muscle. Next, they say describe how the fascia generates a diffuse pain. This is from paragraphs 8 and 9. To detect diffuse pain, pain receptors are going to be involved, and they will detect the stimulus. An impulse is going to be generated, and this is going to be sent through the sensory neuron and transferred to the pain center in the central nervous system for interpretation. Next, they say explain how inflammation in the fascia is caused. This is from paragraph 8 to 9. Inflammation, of course, this is like how we see in immune response. There is going to be histamines, there is going to be vasodilation, more blood to the area, and there is going to be swelling. So I said when damage occurs due to injury or infection, histamines are released, and these lead to vasodilation of the blood vessels in the fascia. So more white blood cells and fluids arrive in the area, causing swelling. Next, I say describe how fibroblasts within the fascia could be transformed into myofibroblasts. This is differentiation, so I said control of gene expression occurs. Transcription factors switch off or on certain genes in the fibroblast. Epigenetic control in which methylation of DNA and histone modification is going to occur. Genes that are activated are transcribed into messenger RNA, and these are going to be translated into proteins. The formed proteins are going to transform the structure as well as the function of the cell, making it differentiate from fibroblasts to myofibroblasts. In part F, explain how lymph is moved in one direction in the lymphatic system. The lymphatic system contains lymph fluid, and the lymphatic system has some valves. There are also some muscles around the vessels, so the muscles are going to contract, and then the fluid is going to move from one section to the next, and the valves are going to prevent the backflow of the fluid. So I said on stimulation by the nervous system, the muscles around the lymph vessels contract. Compressions on the vessels move the lymph in one direction. The lymph vessels also contain valves to control the backflow of the moved lymph. An exercise suggests how aging may have an adverse effect on the structure of the lymphatic system. As somebody ages, the muscles are not going to contract properly, the vessels are going to be weakened, they could be thin, and the movement of the lymph fluid is not going to occur appropriately. So I said the lymph vessels become less elastic and thinner. The endothelial layer may decrease, further weakening the vessels, making them less sensitive to stimulation, and the number of lymph vessels could also decrease. So this brings us to the end of question 8, as well as to the end of the whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.